Hello everyone, thank you for joining this presentation today. My name is Laura Gonzalez and we are going to talk about an algorithm to annotate transposable elements. But first, what are transposable elements? They are DNA sequences with the ability to change their position within the genome, and because of that we also know them as jumping genes. Transposable elements are important because depending on their insertion site, they can affect expression and function of genes near them, and they also play a crucial role in the evolution. Here we have some examples. We have the change of color in the maize kernels, the change of size in tomato, and the change of color in apple. All of them as a result of transposable element insertion. There are different elements and they are commonly classified into families according to the Wicker system. So we have two main classes. The first is known as the retrotransposons or copy and paste, and the second class is the DNA transposons or the cut and paste. Within the families, they can also be classified according to their domain organization. For example, here we have Copia and Gypsy and some difference among them. To annotate these elements, there are different approaches that have been developed. So we have homology-based approaches that use databases to discover the elements based on the similarity of the genome and the element. We have de novo approaches that looks for similar repetitive sequences within the genome, structure-based approaches that use the knowledge of a structure of some of the elements, and the comparative genomic approaches that try to identify patterns or of insertion or deletion of elements within the genome. So the homology-based methods are the most commonly used ones, being repeat masker the reference tool to use. It is also part of a lot of annotation pipelines. However, these tools rely on all versus all comparisons of the genome against each of the elements of the database, and these comparisons are mainly based on BLAST or BLAST-like applications. The problem with these tools is that they require huge computational capacities and time to annotate complex genomes. So we ask ourselves if we can use minimizers that is developing strategy for different genomic approaches and instead of last all versus all comparisons. So first of all, we need to understand what's a minimizer. So a minimizer is a k-mer that can represent a sequence based on a function that is known as the hashing function. That is defined within the algorithm. So as is seen here in this example, we have the sequence S, and we first break it into smaller windows, W. For each window, we pick all the k-mers, and for each k-mer, we apply this function that we define, and then we select the k-mers that got the lowest values of this function. So after that, we can just represent S as this set of minimizers instead of the entire sequence. So in our algorithm, the first step is that we have the genome. So we break it into windows as I previously explained. For each window, we got all the k-mers and we applied a hashing function into them. And we pick the minimizer for each of the windows of the genome that we have as an input. And with that, we build the index of the minimizers where we can store that value of the hashing of the minimizer and its position within the genome. So with that, we can easily identify where is each minimizer located in the genome. In parallel, we have the transposons database or the curated library that we have also as input, and here we break it into k-mers, and for each k-mer we apply the same hashing function that we applied in the genome. The idea here is not to find minimizers, it's just to know with that hashing function where is its location in the database. So after this indexing step, we are going to have a query and the index that are going to be related in terms of the hashing values of that function that we had. So then we need to find the matches of our library against the genome that we need to annotate. So in this case, here we have one chromosome as example and one element, and we have these matching minimizers against the two of them. And so we are going to define a match or an annotation using the first and the last minimizers that are part of the largest match of each of our elements and the genome. So here we have the reported as the first and last positions within the genome that we are annotating, and we are also gonna assign the same family of transposable elements that we got in the library. So we use this algorithm to annotate LTR retro elements, and we compare them against the results of repeat masker because we don't have a gold standard. So here we are assuming repeat masker is the gold standard. So using that, we identify LTR retro elements using a de novo software that is called Impactor 2 for each of the plants that we were analyzing, that are Arabidopsis, coffee, and rice. And as you can see here, we got a higher than 75 of precision and sensitivity using those libraries, and we got a reduction in at least one order of magnitude of our software in comparison to the time that repeat masker requires to annotate the same dataset. We also perform the full transposable element annotation using different databases as the Arabidopsis, coffee databases, the RepBase and the TREP database. And here we can see that if we choose a representative database, we can annotate our genomes with good precision and sensitivity. We can play a little bit with the precision and sensitivity results if we increase the number of iterations of the annotation using the output annotation as the input of the next round. Um, and in all cases, we also got a reduction from 3 to 20 times of the time required to annotate the genomes. So finally, we wanted to know what happened with a complex genome, such as the hexaploid with genome. This genome is important for us because it got an 85% of transposable elements. So it traditionally requires a month to annotate all the genomes, representing a bottleneck in the process of annotation and next analysis. So here with our software, we can annotate the with genome in less than 2 hours per chromosome using less than 250 gigabytes of RAM per chromosome, and we got the same percentages of elements within the genome that are LTR copia, LTR gypsy, and DNA cacta that were previously reported. So, in conclusion, our algorithm outperforms traditional homology-based annotation tools in speed by a factor of 3 to more than 20 times. We expect that these new developments will be very useful for several organisms in studies that are assembling and annotating genomes, and we also expect to make further improvements in the software to increase precision and sensitivity while identifying overlapping elements. So finally, I want to thank you for joining me today. You can read the complete article using this QR or download and try yourself the software in this another QR. Thank you.